Japanese researchers use a quantum circuit to identify single nucleotides, which is a first step toward a quantum computer that can act as a molecular sequencer of DNA. DNA sequencing technology, that is, determining the order of nucleotide bases in a DNA molecule, is central to personalized medicine and disease diagnostics. Yet, even the fastest technologies require hours or days to read a complete sequence. Now, a multi-institutional research team led by the Institute of Scientific and Industrial Research (SANCAN) at Osaka University has developed a technique that could lead to a new paradigm for genomic analysis. Genomic information is essential for human health. Due to its large volume, genomic information can be potentially computed using quantum computers, which are rapidly developing. Genome analysis using quantum computers can accelerate the development of personalized medicine, innovative drugs, and novel diagnostics based on genomic information. However, genomic analysis, including nucleotide identification, has not yet been performed using quantum computers. DNA sequences are sequential arrangements of the nucleotide bases, that is, the 4 letters that encode information invaluable to the proper functioning of an organism. For example, changing the identity of just one nucleotide out of the several billion nucleotide pairs in the human genome can lead to a serious medical condition. The ability to read DNA sequences quickly and reliably is thus essential to some urgent point-of-care decisions, such as how to proceed with a particular chemotherapy treatment. Unfortunately, genome analysis remains challenging for classical computers, and it is in this context that quantum computers show promise. Quantum computers use quantum bits instead of the 0 and 1 of classical computers, facilitating an exponential increase in computational speed. In a study recently published in the Journal of Physical Chemistry B, the researchers aimed to use a quantum computer to distinguish adenosine from the other 3 nucleotide molecules. Using quantum encoding to identify single nucleotide molecules is a necessary first step toward the ultimate goal of DNA sequencing, and it is this problem that the researchers sought to address. The researcher explains that using a quantum circuit, they show how to detect a nucleotide from only the measurement data of a single molecule. This is the first time a quantum computer has been connected to measurement data for a single molecule, and demonstrates the feasibility of using quantum computers in genome analysis. The researchers used electrodes with a nanoscale gap between them to detect single nucleotides. The output of current versus time for the adenosine monophosphate nucleotide differed from that of the other 3 nucleotides. This is because the conduction path of electrons between the nucleotide and the electrodes depends on the chemical architecture of the nucleotide. This is the basis for designing a quantum gate, which serves as a molecular fingerprint for each nucleotide. Variations in conductance depend on molecular rotation patterns that are unique for each nucleotide. In the present setup, discrimination of adenosine monophosphate from the other 3 nucleotides is not necessarily straightforward, but DNA sequencing could be possible by designing quantum gates for these other nucleotides as well. This work has broad and exciting potential applications. Advances in drug discovery, cancer diagnosis, and infectious disease research are a few examples of what is expected with the advent of ultra-fast genome analysis.